Good morning, everybody. Lovely to have so many of you able to join us this morning. This is going to be the first in a series of presentations on medical issues. I'm delighted to have Phil with us. He's very well known to most of you. He is a committee member, and we're delighted to have someone with his medical background part of the team. Phil was educated in Port Elizabeth. I think it was Grey High School, wasn't it? And then you went up to St. John's. Am I right, Phil?
That's right.
So Port Elizabeth in Johannesburg, where he went to school. And then he qualified in medicine at Wits University and studied further at the University of Toronto. He has worked as a specialist radiologist at Groote Schuur and Toronto University, and in New Zealand. So he comes with a very, very profound depth of experience. He has lectured and published extensively in his field. So we are really looking forward to having his insights on the perplexing topic of the moment all over the world, vaccines. We are so fortunate to have someone like Phil to enlighten us. And since he's retired to Hermanus, he has worked in a voluntary capacity at the Hermanus Provincial Hospital. So he's hands-on locally as well. Phil, lovely to have you able to join us today, and we look forward to your talk.
Thanks, Letitia. I must say this has been a very interesting talk to prepare because the ground keeps shifting beneath my feet. Even today, there's new information, important information about the virus and about vaccines. We're in a position now where 2.5 million people have died worldwide from this infection. And in South Africa, we're approaching 50,000 deaths. And that's the same number that we had during the Spanish flu. I think the difference that we've noticed with the second wave now is that we all know somebody who's been critically ill. Poor old Gert had such a hard time. Or even worse, we know people who've died. So this virus is not something to be trifled with. We keep getting news on the TV every day. We've got to be careful. We've got to get vaccinated. And I'm going to just share some of that information with you. What I thought we'd look at initially was just viral infections in the body. You're all aware of many infections. The obviously cold viruses are common, flu viruses in the lung, gut we have to deal with. And the new kid on the block is obviously the SARS coronavirus. So what exactly are viruses? They're very unusual structures. They're not even called organisms. They live at the very edge of life. They are bits of RNA and DNA wrapped in protein, and there are over 6,000 species. They're not living or dead, which is amazing, although you will hear people, medical people, talking about dead viruses or living viruses all the time, but critically speaking, they are neither. You could think of them as a computer virus. So a computer virus exists, but cannot be active unless it's in a computer. And so it would be with an ordinary virus. They are parasitic structures. They need a cell to reproduce, and they take over the cell and replicate very rapidly until they either breach the cell wall or the cell bursts. They have 4 million bases. Now, I gave a talk a year or two ago on human genetics, and we have 3 billion base pairs. So a virus is a very small structure, just 4 million bases. They have between 4 and 200 genes. And the COVID virus has 11. They're called polygenes because they code for more than one protein. So just 11 genes. This is what we're up against. It's an amazing slide I found where you can see the virus replicating within the cell. And here a cloudburst of viruses has been released into the bloodstream. Each little virus here will go and attack another cell and get going again. They come in lots of shapes. This is an Ebola virus, and this is a herpes virus, but they really are tiny. They can't be seen under light microscopy, only with electron microscopy, and they're about 100 times smaller than a bacterium. So here, another picture of the coronavirus. And if you look carefully, you can see little projections coming off the periphery of the virus. And if we look at an artist's impression of the virus, I think you're familiar with these, you'll see that there are many proteins sticking out of the edge of the virus. There is a capsule. COVID virus at least has a single round chromosome made of RNA. Of particular importance are these spike proteins because the virus uses that protein to gain entrance into cells. The other thing about the spike protein is that it is very antigenic. So it creates in the immune system a vigorous antibody response. And because it is so antigenic, it has been targeted by the vaccine companies as the prime molecule of the COVID virus that they try to manufacture. And when we look at RNA, we remember the COVID virus is an RNA virus, not a DNA. You can see that an RNA molecule is a single helix as opposed to the DNA. The RNA viruses still have a language composed of only 4 letters, similar to DNA, but with one difference, that they don't have thymine, they have uracil. And if we look at bits of DNA and bits of messenger RNA here, we can see how things can go wrong. So this is the normal situation with a little piece of a gene, just 9 nucleotides. And when this piece of DNA needs to be replicated, one gets a mirror image of it in messenger RNA. Each 3 nucleotides code for 1 amino acid. So this is arginine, histidine, alanine, and multiple amino acids go on to form a protein. A protein is antigenic and creates antibodies. So what happens if we get a mutation, as you can see here? Instead of guanine, we have adenine in the DNA molecule, and when that is interpreted by messenger RNA, we get uracil, and you can see that these 3 nucleotides code for a different amino acid. So the mutation has caused the production of a different protein. And a different protein may have a different shape. And the different shape can change the behavior of the protein and make the virus different. It can change its behavior. But we'll come back to that in a minute. This is what the spike protein looks like if we make a molecular graphic of it. It's a very complicated structure. So it's made of a protein, and it has a specific shape, and the shape relates to its function. Now we're going to come across words in this talk, and you heard on TV, and I just want to explain what they all mean. We've had a look now at a mutation. Another name for that is a variation. And in October last year, 20 mutations were found in the South African spike protein. And that was in Motherwell, just outside Port Elizabeth. And that made the protein structure change, which caused a behavioral change, which in turn gave rise to a new variant. So a variant or a strain is a virus which has changed behavior. And it changes behavior to improve, teleologically speaking, improve its chances of survival, as we'd expect with Darwinian natural selection. So a virus that has changed to become more infective will become the dominant strain in the population. And we've seen that again with our 501YV2 South African variant. And on the news this morning, America's got a new variant, which they say is worse than ours. Other variants have been found in the USA, Brazil, Denmark. They swap from one country to the next. Now, there's another term you may come across called lineage. And lineage just relates to a variant or the original virus, which breeds true without any mutation. So as soon as another mutation comes in, one would get another lineage. So you can see a lineage is exactly like a clone. A lineage is a line of perfect replicated particles. And look back at what, as you know, we had 3 phases, and I imagine, although there's no documentation because genetics didn't exist at the level it does now, but it seems as if the virus mutated to a much more virulent form between the first and second waves. And they went on to have a third wave, which we will do, and we will go on to have a fourth, maybe a fifth as well. Now, how do we stop the pandemic? There are really only 3 ways. And the first is that the virus can mutate to a more benign form, but so far it hasn't been playing the game. The next way is that we can vaccinate, but we have to vaccinate for all the variants. And that might mean that we need booster shots, which would include the current variant in circulation. You saw that with the AstraZeneca vaccine, which was very good, 70% efficacious for the first variant that we had in this country, before the Motherwell / South African variant arrived. And then we found that the AstraZeneca variant wasn't any good at all. So we have to go on to the next vaccine that is better for our current dominant variant. The other thing we can do is get herd immunity, or as Cyril likes to call it, community immunity. There's a sort of aphorism that no one is safe until everybody is safe. Now, I've spoken to you about an antigen, and specifically the spike protein. And to define an antigen is as follows. It is a molecule capable of causing the immune system to produce antibodies against it. That's all it is. So antigens are usually proteins, not always, but usually. If we look at the structure of antigens and antibodies, antibodies are always depicted as this Y-shaped structure. And the antigen, i.e., which could be the spike protein, for example, or another protein on the virus, have different shapes. The antibody is configured to have the same shape as the appropriate antigen. And the antibody then becomes neutralizing. So you'll understand that if the shape of the antigen changes from this to that, for instance, then the antibody will no longer fit. So the vaccine that we've made may no longer work. It might fit a little bit, but the effectiveness of the vaccine may not be as good as planned. So what happens in the body? We have plasma cells or B cells, B-cell lymphocytes. These have been pre-tuned by vaccination to recognize the virus, the coronavirus. They produce antibodies, the little Y-shaped things, and the neutralizing antibodies then go and attach themselves to the spike protein. Remember, the spike protein is the way the virus enters the cell. And if you block that method of entrance, then the virus can no longer enter the cell. So that's what vaccination is all about. It's priming the body against an impending infection. The immune system is a complicated system that protects us from infections, but not only infections. The immune system is able to determine what is self, what makes up me, and what is foreign material, mostly in the context we're talking about now, infections. The body has a number of barriers. So that we have the skin, we have the lymphatics, and we have structures which contain immune cells. And the oropharynx has the tonsils and the adenoids, which are the first to catch hold of bugs coming inside us. And other parts that make up the immune system are the thymus, the spleen. In the intestine, we have little things called Peyer's patches, which are collections of immune cells just beneath the mucosa. And then we have a very rich complex of lymphatics all around the body, particularly in the pelvis here. The other part of it is the bone marrow. When we look at the immune system on a cellular level, we have the myeloid progenitors, it's called. The initial cells father all these other little cells, neutrophils, basophils, eosinophils. And then the other part of the cell system is the lymphoid cells. And these are killer cells and B cells. The B cells create the little antibodies that we've been talking about. And when the body is exposed to an antigen, memory cells remember what the antigen looks like and are able to mount the attack should the virus come into the body. So when you go for a white cell count or a blood count with your doctor, generally they measure these cells, and this would be a white cell count. And then they can do what's called a differential, and then they would look at each one of these cells individually. Neutrophils increase with an infection, eosinophils increase with allergies, etc. Now, there are two cells at the bottom here which are very important. One, but this is an antigen-presenting cell, a dendritic cell here. This little cell we're going to come across frequently when we get to vaccine production. The other important cell here is a macrophage, and macrophages are cells which gobble up infective agents that come into the bloodstream. So when we look at immunity, there are two parts of it. We have the innate response. So this is our generalized weapon system. And if a virus comes into our body or a bacterium, macrophages gobble up whatever it is and destroy it. Basically, they break it up into pieces. And this happens, this response to neutralize invading organisms happens within hours. What happens then is the broken bits of the virus get presented by the APC, the antigen-presenting cells, or the dendritic cells that I showed you on the previous slide, these guys down here. And the antigen, i.e. the spike protein, is presented to cells, which then remember what it looks like, and they're presented to B cells, which create the antibodies. So when we get exposed to a virus, we have the innate immune response, which kicks off and starts gobbling up the virus, getting rid of it from our body. But what usually happens is there aren't enough cells to complete the job. So then the virus gets the better of things, and we get an increased viral load. We start getting symptomatic, we start getting sick. And then the next thing that happens is the adaptive immune response comes into play and the virus gets killed off, hopefully. So to go through this again, because we're going to come to this with vaccines, we have the macrophage gobbling the virus, breaking it up, presenting bits of it, and in this case, we're going to be talking about the spike protein mostly, to lymphocytes. Lymphocytes then become either killer cells or plasma cells, which are able to create antibodies, and memory cells are also created. How do vaccines work? They work by introducing weak or inactive forms of the disease into the body. The body reacts by stimulating the immune system to create antibodies. And then the antibodies remember the disease and can defend against it if a person is exposed. How are vaccines made? Things have changed. In more recent years, we've gone on to reverse tech vaccinology, which I'll tell you about now. But traditionally, vaccines were grown, the virus was grown in eggs or bacteria or yeast cells or even plants, and were then fermented and the vaccine created. Modern vaccines start with the genome of the organism. So we go back to that single chromosome, and from the chromosome, the relevant sequence for the protein, the relevant gene, is isolated. Once you have the gene, you know what the protein is, and the protein is the antigen. I hope I've explained that. There are many phases in making traditional vaccines. It's a long process. Before coronavirus came along, the shortest duration for making a vaccine was 4 years, and that was in making the mumps vaccine. But the virus has to be cultured, harvested, purified, inactivated, formulated. Vials have to be filled. They have to do freeze drying, packaging, release the batch and retest, and then transport. So in looking at the vaccine candidates that we have now, how are we going to get immunity to the coronavirus? We can get infected, as poor old Gert did, by the community with the virus. And in that way, we create spike proteins, which in turn create antibodies. Or that can be done artificially. And there are 4 ways to do that. We'll just have a look at those quickly. Using dead viruses, using nucleic acid, using viral vectors, or putting in the protein ab initio into the body. So the traditional vaccine, this is what they used. They cultured the virus in eggs. This still happens. It's a very effective form of making vaccines. One of the problems with it is if you have to make billions of vaccines, you need billions of eggs, and you can run out of eggs. But it's very effective. So the first way of making vaccines is to make an inactivated or attenuated live virus. And this is the traditional old way of doing vaccines. It's the one that's used for measles, mumps, rubella, polio, flu, and COVID. And the vaccine that's currently using this technology is CoronaVac. Not many people are using a live virus. They're rather using an inactivated dead virus. The idea, again, is to inject the viral particle into the body. That viral particle, again, gets gobbled up and broken down by antigen-presenting cells. So the little peptide, the little protein molecule of the spike gets presented and the immune response follows. I thought we'd just go back and look at the structure again of the virus. So we have spike proteins, we have a single ring RNA chromosome, and this chromosome contains 11 genes, and it is possible to cut out one of the genes, i.e. the one that codes for the spike protein, and to make a vaccine from that. So with genetic engineering, this is what they do. They cut out the appropriate piece of the RNA that codes for the spike protein. And they then splice that into the genetic material of a neutral virus. Now, Last time I spoke about this, I spoke about the use of chimpanzee adenoviruses, and Gert was worried that we might turn him into a chimp, but no chance of that, because everything happens outside the nucleus of our cells. So the spliced gene protein is injected into us, or another form of this is to have a non-replicating viral vector where the genes that allow the virus to replicate are destroyed. In both instances, viruses are gain access to the body via injection, and the same thing happens. They're gobbled by macrophages. Macrophage then turns into an antigen-presenting cell. The spike protein is then presented to the immune system, which forms antibodies. You will notice from this technology that if you can splice in one gene, it wouldn't be difficult to splice in two, in theory. So you can use this technology for inducing immune responses to pathogens, or you can use it for vaccinating against two strains of the virus. And this is being done by a couple of companies. We'll come to that later. The third way of vaccinating is to use the spike protein as a substance. So you inject into the muscle of the arm spike protein. The same thing happens. Spike protein gets gobbled up by macrophages. Macrophages turn into antigen-presenting cells. The spike gets presented and an immune response follows. Now, the final way just needs a bit more explanation, and that is the RNA viruses. So this cell shows you how proteins are made. DNA unravels in the section that is relevant to the production of the required protein. A bit of RNA, called messenger RNA, then creates a mirror image of the DNA code. That RNA travels through the wall of the nucleus, and in the cytoplasm of the cells, it presents itself for amino acid production. So what happens, as you saw before, that a set of 3 nucleotides equals 1 amino acid. And as the messenger RNA is read from one end to the other, so the chain of amino acids grows till eventually it forms a protein. An RNA vaccine consists of a messenger RNA strand that codes for a specific disease antigen, in this case, the spike protein. Once the messenger RNA strand in the vaccine is inside the body's cells, the cell uses that genetic information to produce an antigen. This antigen is displayed on the cell surface where it is recognized by the immune system, and antibodies are produced. So here we go. So won't worry about the left-hand side of this slide. That's the DNA. We're going to just worry about the RNA vaccine. So what they do is they isolate a piece of RNA which contains the genetic material for the spike protein. That's injected in the cytoplasm of the cell. Spike proteins are made, go to the APC, go to the immune response. The RNA vaccines are a way of shortcutting many of the other methods of producing antigens. Their advantage is that they're relatively easy to construct, and they're very quick to make. So that when the original sequence of the coronavirus was given out by the Chinese, within a week, Moderna and Pfizer had created artificially the information for the spike protein. If we look at the COVID virus again now, from what I've told you, the spike protein is a very complex structure. and as with all viruses, is particularly prone to mutation. So as we've seen now to our disadvantage, multiple mutations in the spike protein have led to resistance of the currently marketed vaccine. So a way around this is to use more than one antigen. And there's a company doing that now. The information just came out last week about that. It's a company that's involved with UCT, and they are using the spike protein and the nucleocapsid protein, a little bit of protein that the RNA is wrapped around. So that is called a second generation vaccine, and watch this space. When vaccines are made, they have to go through trials, and you will have heard of phase 1, phase 2, phase 3 trials. The difference between the different trials is the number of people involved in these trials. All of these trials look at the safety of the vaccine, the side effects, and the effectiveness. It's not without risk, I might say. You know, people have died in phase 1 trials. They've had bad complications. Any side effect causes the vaccine to be pulled off the market, either permanently or until they can sort out the problem. How good is a vaccine when they make it? They look at the likelihood. Does it prevent infection? Does it reduce the severity of an infection? And does it reduce the likelihood of transmission from one person to the next? There are a few examples here of viruses that have been used. Shingles vaccines are very effective. There's one thing we have to just remember, that in older people, in the elderly, i.e. people over 65, the effectiveness of vaccines is diminished. Why? Because our immune systems don't work as well as they used to when we were younger. The other thing you need to note is that when they tell us that a vaccine has 90% efficacy, that presumes that it doesn't work in 10% of people who have it. So these are not, although we have to have vaccines and although they are likely to help us, they may not. But shingles is a good one. Seasonal flu, as we all know, works sometimes and sometimes doesn't. Polio vaccine is good, and polio is only present in the wild in Afghanistan and Pakistan at the moment. Otherwise, it's been wiped off the face of the earth, and hopefully it will. And then there's the COVID vaccines, which we'll have a look at now. And the minimum that's required for a vaccine to be thought of as effective is only 50%. So it only has to work in 50% of people for the World Health Organization to call it a success. A perfect vaccine would look like this. It would be safe. It would have long-lasting immunity. It would provide good protection against all variants. It would induce a response that protects against the disease. It would rapidly induce immunity. It would be simple and cheap and would be effective for all individuals, the old and the young and the ones with comorbidities. And it would require few immunizations to cause protection. So the sad thing is that the perfect vaccine doesn't exist and will never exist, but we get good vaccines. The other thing we've got to look at, of course, is herd immunity, which we've heard a lot about. Herd immunity is the indirect protection from infection of susceptible members of a population. And it's brought about by the presence of post-infective or vaccinated immune individuals. So the more people that are immune, it makes perfect sense, the less likely you are to catch the bug from them. That's the theory. It is the depletion of susceptible hosts in a population. It's like creating a firebreak. And the population will be protected when herd immunity is 70 to 85%. So let's look at what's on the market. At the moment, there are approximately, this changes from day to day, 37 products which are in trials. The ones we heard a lot about are the Rna vaccines, one, the Moderna one produced in America and the Pfizer in Germany. They are very efficacious. Efficacious is not the same as effective. Efficacious is a word used in trials, and effective is looking at the infection in the wild. Generally speaking, the effectiveness of a vaccine is less than its efficacy. The other one that turned out to be a bit of a damn squib because of our variant was to AstraZeneca, but it is being given out in, I think, Ghana now and Pakistan, other countries. And it will, of course, not be efficacious for our strain, only 20%. But that's a vector-type vaccine, the same as the Johnson & Johnson, which is going to be offered to my sister-in-law, who's a GP, is getting her vaccination on Friday. That's 70% efficacious single dose. The Russian virus is a good one. It uses 2 vectors. And the first shot gives you 70% protection, the next 91%. Rna Chinese, which we spoke about, was the inactive virus. Covishield is just the Indian version of the AstraZeneca. There's another mRNA one, which is made in Germany, but it's still in trials. A German one which uses the protein way of making things, and then this final one made an American company, Immunity Bio, which is second generation, the first second generation vaccine. It's a vector vaccine, and that's going to take about a year to develop. I've made a whole lot of questions here, so we can just go through them. Right, now, is a variant more infectious, more deadly? It may be. Our South African variant is more infectious, but it isn't more deadly. There are 2 new variants disclosed today on the news in America, one on the East Coast, one on the West Coast. They both seem to be more infectious and more deadly. And they also seem to be resistant, partially resistant to the Moderna vaccine, the Rna vaccine, which they've created. So that's going to cause problems for them. And eventually it may cause problems for us because the nature of viruses is they spread around the world. So people who've been vaccinated in the States may need a 3rd vaccination to cope with the next variation. Am I immune after infection or after vaccination? Not always, and not totally. It depends how badly you got infected. So Harries, who had a very bad infection, is likely to have a lot of antibodies. Is he immune? He will be a month or 6 weeks after he had the infection. But is he immune to all coming variations? No, he's not. And he may have relative immunity, but it may not be total. How long does immunity last? That's an unknown question. It seems that immunity lasts 6 months at least, maybe longer, but we don't know. Infection has not been around long enough. Can you get infected after vaccination? Yes, you can. Because if another variant comes along, you can definitely get infected. And so far, there have been 4,000 documented cases in South Africa, not after vaccination, after infection. But the same holds true. Can you get infected after vaccination? Yes, you can. Can you get reinfected after being infected? Yes, you can. Transmission after infection or vaccination, we don't really know about that, how good the vaccines are at preventing transmission. And that, of course, would affect herd immunity. Is there going to be ongoing viral mutation? absolutely there is. And until we come back to that maximum, until everybody's safe, no one is safe. So while the Americans are busy vaccinating themselves and protecting their community, or the British doing a better job of it, here in Africa, the virus is going boss, and we will continue to have mutations. Those mutations may spread to other countries. Are we going to have a third wave? Definitely, and maybe more. Why should we get vaccinated? Well, you will diminish your chances of getting severe disease or of dying. So if Harries had been vaccinated, in theory, he would not have had such a bad time of it. We are going to see, as I described there with the vector vaccines, multi-strain vaccinations, so that there will be quadrivalent vaccines, which will deal with multiple strains in one go. Why are so many companies making vaccines? Well, I saw in Daily Maverick today that Moderna says they're going to make $18 billion of, I don't know if it's profit, but $18 billion of their vaccine. Do I need a second injection? Definitely. If you're not having the Johnson & Johnson vaccination, all of the others need 2 injections. So you have one which starts off the immune system, the next one boosts it and strengthens it. You need both. And can you mix and match vaccines? In other words, could I use an RNA vaccine and then go on to Johnson & Johnson? That used to be an absolute no-no, that you could never, ever do that. But now it is accepted that you can. Now, in looking at the virus, two weeks ago, Kingsbury Hospital in Cape Town had a little morning conference on personal experiences by doctors. And a new syndrome, which I've never heard of, has arisen, and it's called moral injury. And it's an injury to an individual's moral conscience and values, which produces profound emotional guilt and shame with transgression of one's own moral beliefs, values, or ethical codes of conduct. It seems that the health community is getting so battered by this blooming virus and in hospitals that psychologically a lot of them are in turmoil. They had a number of doctors speaking. At the end of their 10-minute talks, 2 anesthetists and 1 physician were in tears. Very difficult to watch. And why is that? Because they're making decisions like who gets the bed. Harries experienced this. Who gets the oxygen? If you have a premature baby and an old man with COVID and a post-operative patient, and you have a limited oxygen supply, who gets the oxygen? Who gets the ventilator? Who's allowed to visit? Well, you know, nobody's allowed to visit patients with COVID, and patients are dying alone without family support. And then finally, you know, there's going to be this question of who gets the vaccine. And again, this morning I read that the politically connected are bypassing the rules that we have in place for medical workers to get the vaccine initially, and they are now ahead of the queue. And we've seen, you know, in South Africa, I was astounded to see this picture in the Daily Maverick of this was the AstraZeneca virus arriving on a Friday afternoon, I think. And the police guard that came with the vaccine was astounding, just so it wouldn't get stolen, which probably it would have if they hadn't all been there. And I believe the next one, the Johnson & Johnson, had the protection of the army. So what's in the future for us? It's really much of the same. And we're going to have to keep our distance, keep ventilating, keep the place ventilated. We're going to have to wash our hands, use sanitation, and we're going to have to wear masks. And I hope they don't have anything else in store for us. There we go. Thank you.
Thank you very, very much for an amazing presentation. Can I quickly jump into the queue? I know there are many questions. Is my infection equivalent to a vaccination? And do I need to
Yes.
be vaccinated again now that I've had the illness?
If we get more variants, you will need to get vaccinated again. How good are your antibodies? I don't know. You could go and have that tested to see if you are immune. But remember, you're immune probably to the 501Y variant. That's the likely one that you had. I did ask a vaccinologist last week whether they can tell from the blood test which variant you had. And the answer to that is no, not at the moment. So will you need a vaccination? If I was you, I would. You're not going to lose anything, and you're just going to get more antibodies. You're not going to be immune till 4 to 6 weeks after your last symptoms. I don't know how long it's been now.
It's almost 6 weeks now since my very first symptom.
Is it? So,
yeah,
the chances of you getting it again are slim, but it's possible. And if you get it again, you will get it very mildly.
That's fine, thank you. Questions? There are lots of questions.
There's a question from Peter van Niekerk, Phil. Why didn't we use the AstraZeneca vaccine? Wouldn't it at least have protected us against the old COVID variant, if and when it comes back?
Well, if and when it comes back, I don't think it's going to come back because the virus has mutated and we now have a virus which has advantages over the original one. So this is going to be the predominant variant until another one arrives. But is the old one still out there, alive and well? Yes, it is. But it's only about 10% currently, if not less. So it seems to be fading out. Why didn't we use it? I mean, the fundis said if we'd used AstraZeneca, it would have protected us against what you had, or that was the initial thoughts, that it would protect against severe disease or death. But as things progressed, it seemed that the chances of getting that protection were less and less. Remember, it went from 70% efficacious down to 22%, and then some people were talking sort of 0. So to the point that in Germany now, there are almost riots against having the AstraZeneca vaccine. As far as the population of Germany is concerned, it's useless. And until they change their formulation, they won't use it. So.
A comment from Anina that was quite interesting. She says, it's interesting that we are all happy to go for a genetically engineered vaccine, but there are many people who have uninformed resistance to GM foods.
Yeah.
So that is interesting.
That's very good. The other thing I didn't comment on, that with the viral vector vaccines, they're putting this little piece of the COVID vaccine into the genetic material of an adenovirus, perhaps one that isn't so infective. We may have immunity. We may have been exposed to that adenovirus before, and we may have immunity against that virus, right? The one that they're putting the extra bit of RNA in. So it may not work. That's another reason for them not to work. It's a small percentage, but it's a reason.
Well, as I experienced, the main effect of the coronavirus infection is that it affects your lungs, and therefore you need oxygen. And now that I'm told that I'm in the recuperative phase, I need to watch out that I don't stress my heart because there isn't enough oxygen necessarily in my lungs. But now, with the various mutations, are other parts of the body affected, or does it still go for the lungs?
No, that's a difficult question. It goes for more than the lungs. It can leave kidney damage. It can cause, I don't want to, this didn't happen in you, so I don't want to imply that this is going to happen in any way, because I don't think it is. But when you're very sick, it affects the clotting mechanism. It can give organ failure of various kinds, kidney failure. It can give heart attacks. It can give strokes. it can give all sorts of things, but that's when you're in hospital and you're desperately ill. That's why people die. Now you're in the recovery phase, and I think what's happened, your lung function is diminished. You're still getting short of breath when you exercise. Has your vascular bed in the lungs been affected? I don't know, but it's possible that may be one of the reasons why you can't why your exercise tolerance is diminished. Sorry, did I answer that question?
Oh, you did. Thank you very much. So it really depends on the individual. It can differ from individual to individual.
Very much so. Very much so. Yeah, and I don't know why. Yeah. I mean, you're in one family. You can have two people that get desperately ill and two people who don't get it, so some people are going to be asymptomatic but still have the virus, whereas some people are going to manifest severe disease. It's very strange. Yeah.
Well, is it true that blood type O persons are less susceptible to the infection than other types?
I don't think so, Gert. I haven't heard that. I would think that...
There was an article about it a couple of weeks ago.
Where, in what? Where did you follow the source?
I heard it on CNN, I think.
Did you? Okay. I haven't.
I don't know where Gert heard it, but I did hear it about 2 weeks ago.
Okay. I haven't heard that. None of the talks I've listened to have mentioned that, but maybe. Yeah.
Well, Carol was not affected, and she's got blood type O. So there's one possible proof.
Okay.
How did she not even test positive? I know she didn't show symptoms.
No,
but
she did not. She did not test positive at all. She tested negative.
Amazing, absolutely amazing.
There are more questions and answers, aren't there? Always with this thing, and it continues to surprise and to change all the time. Yes. Robin's saying with all the mutations, it looks as if lockdown may persist. Well, it could well. Yeah, sure, why not? The variants in America that they spoke of today, you'll see that on CNN today, Letitia.
Yes, in Los Angeles and New York.
Are worrying. They are worrying because they are similar, but not the same as the London and South African, the English and South African variants, and they are worse from what they said. They're more infectious, and they are more deadly. So it sounds as if they're in for another wave, and with that we may be too.
Another interesting comment on one of the newscasts, that the longer it takes for a population to reach herd immunity in a country, the more mutations are likely to occur. Correct. Because every time a virus produces something, there's a slight change that happens until it results in a final mutation. Therefore, the longer it takes for us to reach herd immunity, therefore the more and more mutations will arrive.
Yeah, that's correct. I mean, that's why I say, so the Americans and English are busily vaccinating their populations, but this is a problem which involves the world, not just them. And they have to look after us as much as they have to look after them, because we are going to carry on making mutations in Africa. And the virus may die out if we get a good mutation, but so far it hasn't been behaving.
It's got to become a global focus.
Exactly. That's what the World Health Organization says. It is a global problem.
First of all, Phil, an excellent job at taking a very, very difficult subject and putting it simply, and even I could understand it. I was very, very good. I think that one thing that people must understand about the mutations that take place in the new variants, as Phil said right at the beginning, viruses are neither dead nor alive. They are only alive when they're in you, when they're actually working. And so they can only mutate while you are infected. And so the more people that are infected, the more likely you are to get mutations. And so it's a double-edged sword in a way, is that not only are you less, if you cut down the number of people who get infected, you obviously cut down the number of people who are going to be infectious, but also you reduce the time that the virus is sitting in your body, working away and mutating. And so it's like a double-edged sword that the two areas have to be thought of. And I think that's what makes the neglect in the USA, for example, where the pandemic really was allowed to get out of hand for all sorts of reasons, and it's true all over the world, is that when people are infectious, mutations will happen. When there are less people infected, mutations will fall off gradually, and eventually we'll overcome it.
Thanks.
Thanks, Patrick. Well, that's a positive projection for us to look forward to. But I think, Phil, we've got to take your point that the precautions we have got used to exercising, we've got to continue to do so.
Yeah.
And that's the important message.
One interesting, you can see how effective they are, because how many of us have had the seasonal flu this year? Well, I got vaccinated, but even so, I can tell you the pharmacy is selling no flu meds.
No.
I asked them. And why? Because we've improved our hygiene, haven't we?
Mm.
And now, I hope we've all learned a lesson from this. We have, that we must wash our hands all the time. We come home from the shop, whatever, we mustn't touch our face without, we must wash our hands all the time, sanitize, etc.
Mm. Yeah, we've learned very important lessons. And what I think has been important this morning, we've got our medical experts. Patrick, thank you for your input as well. But Phil, a very, very interesting and wide-ranging and really insightful talk. And you've been able to use Gert as a case study.
Yeah.
So it has worked extremely well. And all we can look forward to is for improvements through vaccine and, of course, our behavior. But this has been so interesting, and also you've given us warnings about don't relax, don't think it's all going to go away, and prepare for the worst and hope for the best, which is my little motto at the moment. Thank you so much. And thank you, Gert. I think it's just wonderful that we've had Gert able to be our host today.
Yeah, absolutely.
The example of what is possible with the miracles of modern science.
Goodbye, everybody.
Thank you.
Excellent. Bye.